presentation of panelists today is actually a reproduction of our GSA symposia that we did. And I really feel like so much of what was presented is relevant to folks in demography and also a lot of work that's being done in NCHAP. So I wanted to have everybody come here. So I'm really, really grateful to the panelists and Jen, our discussant, for being willing to come back and do 2.0 on this symposia. So we're going to talk about some emerging biotechnology markers of cognitive impairment today. So just a little bit of background for framing, especially in this group who may or may not have studied cognitive impairment in the past. Cognitive impairment is very, very, very prevalent in the US right now among 65 and older adults. At least 1 in 9 older adults have some degree of dementia, and this is more highly prevalent among older adults in minority racial ethnic groups. So it's really important for a number of reasons, not just with the aging of our society, but also because of the racial ethnic disparities that come along with it. At the moment, we have not great pharmacotherapy to treat dementia, and so a huge emphasis remains on prevention through things like physical activity and diet and smoking cessation and prevention of stroke. DNIH continues to fund a lot of pharmacotherapy development research, but we just don't have a lot of great agents yet. So there's really important, I think, space in a clinical setting for early detection of decline and also potentially prevention of additional decline. With that cognitive background, we have these now extremely available wearable sensors exploding on the commercial markets all over the research space. And these wearable sensors allow us to continuously collect this remote movement data in free living spaces. It's passively collected while people just go about their life. It really is low burden on the data collection side. And it's extremely granular movement data, even up to 100 or more points every second. And this kind of wearable sensors really offer the opportunity for new biometric markers of health. What's interesting, though, is that a lot of this work has been done in frailty and in cardiovascular disease and in readmission and recovery, but not a lot has been done in its association with cognitive function, even though mobility and physical activity is a huge preventive measure for cognitive decline. So today we are fortunate to have 4 unique presentations about biometrics and their association with cognitive impairment. And we have co-authors that are representing 7 different institutions. So this is as fun as team science gets. What's also very interesting about our presentations today is we're going to see some evidence of wearable sensor data and non-wearable sensor data across different body locations and different types of sensors. So we've got 3 different types of body locations represented, as well as some in-home motion sensors. So our first presentation is going to be from Dr. Teresa Tian. She is from the National Institute on Aging, and she will be talking to us about a relatively new metric, which is a relative vigorous intensity physical activity, as opposed to just vigorous physical activity intensity, and its association with imaging, brain imaging, and microstructural changes in the brain. Dr. Amal Wanigatunga is going to talk to us about physical activity patterns across the day and its association with longitudinal changes in cognitive decline in the Baltimore Longitudinal Study of Aging. Dr. Jill Rabinowitz is going to look at the 24-hour activity rest circadian pattern and relate that to cognitive decline in the same data set. Chengjian, who is a PhD candidate here at the University of Chicago, is going to look at some hip accelerometry data in the free living environment and its ability to predict cognitive decline among older adults, predominantly minority older adults, who are do not have cognitive impairment at baseline. And finally, we will have a really unique presentation relating the room-to-room transition, like in-home room-to-room transition to mild cognitive impairment in older adults from Dr. Wu, who's going to join us at around 1 o'clock. And finally, Dr. Jen Schrack, who is an absolute guru in accelerometry analyses, international leader in this space. We're really fortunate to have her as a discussant and to kind of help us talk a little bit about the implications of these findings and what's next in the future. So I'm going to stop and have everyone put their questions in the chat or just unmute yourselves. We have a small enough group, I think we can do that. And I will let our first discussant share her screen. Dr. Tian? Great.
Okay, so can you see my slides okay?
Yes, they look great.
Great. Okay, so I get started. My name is Teresa Tian. I'm a staff scientist epidemiologist from the National Institute on Aging. And thank you, Megan, for the nice introduction. It's a great pleasure to be here. So today I will be talking about a study, not quite from the detection perspective, but from the perspective of a modifiable risk factor. So the title for my talk is Longitudinal Associations of Absolute versus Relative Vigorous Intensity with Brain Microstructural Decline in Aging. I have no commercial relationships to disclose. So I'd like to start with a brief background. Over the past decade also in the literature, it has been a very consistent finding that being physically active or more participation in physical activity can protect against neurodegeneration. And this has been supported by neuroimaging studies. And the findings has been mostly cross-sectional. So I think the first gap in knowledge here is there's a need to understand the longitudinal data because the cross-sectional association does not necessarily reflect the within individual decline over time. Another gap in knowledge is that most of the previous imaging studies focused on the volumetric measure, which is conventional and looking at how big the brain volume is. But the data on the microstructural change is very limited. So it is important to understand the microstructural integrity because the microstructure via the diffusion tensor imaging tracks the directionality and the diffusion of the water molecules in the brain. So it may be more sensitive to detect the early change with aging compared to the conventional volumetric measures. Another very interesting observation that I noticed in the literature is that what is the specific dose? For example, at what intensity level may be more strongly associated with the neuroimaging markers of interest? And the findings are not quite conclusive. There has been some recent study that look at the specific intensity level. So for example, they suggest that moderate vigorous intensity activity is more strongly associated with the brain structure compared to light intensity activity. But the findings has been not consistent overall. So I think one reason could explain the discrepancy is that there are different ways to quantify the activity intensity. So for example, it can be quantified as absolute thresholds at the population level, or it can be quantified as relative thresholds at individual level. So the goal of this study is to examine the longitudinal association in the microstructural integrity of both the gray matter and the white matter in the brain. And I'm interested to look at the association with both absolute versus relative vigorous intensity level. And here I focused on a sample of well-functioning older individual without initial cognitive impairment and aged 50 and older. So our hypothesis is that higher levels of vigorous intensity would be associated with a less decline of microstructural integrity over time. And I hypothesize this association would be present in both gray matter and white matter. And because for older individuals, their physical function is important for them to decide how safely they can engage in physical activity. So I think that in this well-functioning population, we hypothesize that the association would be independent of initial physical function. So we used the data from the Baltimore longitudinal study of aging, and we identified 260 participants, and they're aged 50 and older at the baseline. And we had a couple of inclusion criteria to start. So they are required to be cognitive normal at the baseline. This is based on the rigorous adjudication of cognitive impairment or dementia. And we also require the participant to be well-functioning at baseline. So this is defined by the gait speed at least 1 meter per second. And these 260 participants, they had the baseline data on the physical activity measured by the active heart accelerometer, and also the diffusion tensor imaging during the follow-up. The average follow-up here is about 3.8 years. And this is quantified by the 3T diffusion tensor imaging. So the participants have the repeated imaging over time. So regarding the activity intensity measure, as I mentioned earlier, there are two ways to quantify. So one is we refer here as the absolute threshold, which is population level. So for example, here you can see the different cutoffs to define sedentary, light, moderate to vigorous intensity. And for the relative intensity cutoff, here is defined by the individual heart rate reserve. And with a different percentage, it can be quantified into different levels. So in this study, we focused on the vigorous intensity as supported by the previous literature. So regarding the neuroimaging marker of interest, again, we focused on both gray matter and white matter. And we focused on several regions of interest important for different function. Here, for example, we look at several temporal regions, and those are important for memory. And we also look at the prefrontal and superior longitudinal fasciculus. Those are important for executive function, processing speed. And we also look at several motor-related regions, including the primary motor area, the putamen caudate, as well as the white matter body of the corpus callosum. So the statistical analysis has been straightforward. We use the linear mixed effects model to look at the longitudinal association. So in this model setup here, the parameter MVPA stands for the moderate to vigorous physical activity times interval here represent the association between the baseline activity with a change in the microstructural integrity over time. And we considered basic demographics as the covariates. We also include education, which is important for brain health, and also we include the ApoE4 carrier status. And as I hypothesized earlier, we adjusted for gait speed as a measure of physical function. And because we look at a couple of regions of interest, so we look at multiple relationships. Here we set the significance at p now less than or equal to 0.01 as the significance level. So here's the summary of the associations. And here is a list of regions of interest important for memory, which is mostly localized to the temporal area. So as you can see that we did not find significant cross-sectional association with either relative or absolute vigorous intensity activity. However, it is very important to note that we see the longitudinal association with the relative moderate to vigorous intensity levels, which is defined by the individual-based heart rate reserve. And the regions are localized to the temporal region. And we did not find significant longitudinal association with absolute vigorous intensity, which is based on the population level. And to be more intuitive, here I include a summary of the results here for the locations of the regions we find that are associated with relative vigorous intensity. So here, the red is tensor fasciculus, which is around the temporal area. And we also see the association in the medial temporal lobe, including the internal cortex and parahypogyrus, as well as the white matter, which is the hippocampal part of the cingulum, indicated in the yellow here. And when we look at other regions of interest important for executive function, processing speed, or motor function, we did not find significant either cross-sectional or longitudinal associations. So to summarize, I think that the findings clearly show that each standard deviation, more time spent in the moderate to vigorous physical activity, which is equivalent to 21 minutes per day, is significantly associated with less decline in the microstructural integrity. And we found this association is localized to the temporal areas, and including the 4 regions I listed above. And we did not find significant association with absolute intensity level, which is defined by the population-based threshold. And we did not find a significant cross-sectional association. I think part of the reason here may be because we look at a homogeneous sample. Everybody is well functioning to start, and they are free of cognitive impairment at the baseline. So regarding the research implications, so for the public health implication, I think more time spent in activity at least 60% or above of individual heart rate reserve would be associated with a less microstructural decline. And using the absolute threshold does not predict future change in microstructural integrity. So I think future intervention studies may be considering using the individual thresholds to define intensity level to describe the physical activity description among other individuals. I'd like to acknowledge that study is supported by the intramural research program of the NIA, and I would acknowledge the study participants and staff. So currently the study is under revise and resubmit. And if you have any questions, I'd happy to answer now, or you can contact me via emails. Thank you.
Thank you so much, Dr. Tian. I actually think we have time for one question before we go on to our next speaker. Or we can just wait and talk about all these at the end. Does anyone have a question right now? Okay. Oh, Lena.
So if you're an individual and you want to improve your chances, your well-being, you have to wear a heart rate monitor basically, and think about your activity that way?
Yeah, I think, yeah, based on the finding here, I think you need to track the heart rate, like while you're exercising, right? So you could not think of other way. Maybe Jen could chime in.
So I think what's important here is that as people get older, relative versus absolute intensity can mean very different things. So if we held everyone to the same threshold, whether they were 65 or 85, we might see different things. And so Teresa was saying, actually here we didn't see anything because people are pretty well functioning. But if you compare, a general 65-year-old to an 85-year-old and expect them to meet the same guidelines, it's not really feasible. But if you say, try to be at about 60% or greater of your heart rate, your capacity, that does seem to matter. So it is kind of, it's important to look at individualized estimates when thinking about people in older adult age range.
I did have one additional question to that. If you exercise more, then your capacity increases. So if you, the same exercise over time, if you do it a lot, will use less of your reserve. Right? So is this then a ratcheting effect, so that if I exercise for a year or 2 years at one level, in order to maintain the same relative change, I'll need to exercise more? Because once I've trained for a year, going at a particular pace no longer uses my heart reserve. So does this mean I should be does this mean that you're doomed to continue to exercise more and more and more until finally it kills you, or is there a better plan?
That's a great point. I think this can go to the circular. As you said, more exercise, the heart rate reserve gets better. Again, this is an epidemiological observational study, right? I think maybe intervention studies could answer your question, and we could go into different levels while the intervention proceed.
I think the important point is, like, if you're exercising, if you're always exercising, you're not going to lose capacity, right? You might become more efficient at that individual task, so it doesn't push you as hard. Like if you're a runner and you run every day and you run on a flat surface, and then one day you run hills, that's going to push you more, right? Whereas if you run flat every day, you're not going to lose fitness so much as you're just going to become more efficient. So I don't know. It's an interesting question, but I think you'd really have to follow people for like 20 years to understand if that's possible or not.
Sure, I agree. But it does seem, since we've always believed that exercise improves your capacity, and therefore what used to be a struggle when you started is no longer a struggle after 6 months, and therefore we tend to do more. But it does suggest, if it's really relative, that it needs to be calibrated by whether you're actually also changing your reserve, right? It seems unreasonable to think that you're going to have to exercise more and more the fitter you get, right?
Right. That's not the message we want to convey here, right? We want to keep your capacity higher, right? And as long as you don't step it back, I mean, you're going to lose capacity as you get older, no matter what. But if you
I don't think that's true.
Active. If you keep active,
exercising.
But I know we have to keep going, so I'm gonna.
Thank you very much.
No, I actually appreciate this. And, as people get older, it gets harder and harder to exercise anyway. So even if you don't run hills over time, it's just that flat surface is going to get harder and harder. Thank you so much, Dr. Tian. I'm going to put, there's one comment in the chat for you. I'm going to have you take a look at that. And then I'm about to put a question in the chat for you as well. Let's get started with Dr. Wanigatunga though next.
Sure. Hi, everybody. Thanks for having me. Can everyone hear me? All right, let me share my screen. Do you see the full screen of my, okay, perfect. Thank you for having me. I'm an epidemiologist at the Johns Hopkins School of Public Health. I'm in Jen Schrack's working group, and we do a lot with accelerometry. And today I'm focusing on our results from the BLSA. And we'd like to also not only think about physical activity as this modifiable type behavior to help prevent or slow declines in cognition or prevent dementia, but also maybe even think about physical activity as a surveillance tool. And the way we think about it is when you're awake and you're doing your day-to-day activities, you are in some level of physical activity. So here we're kind of flipping the script a bit, and we're thinking about cognitive diagnoses and whether there's differences in these everyday patterns of physical activity, not necessarily exercise, but going from room to room or going to your meetings or whatnot. So thinking about activity in that regard. Okay, so I have no disclosures to report. So really the intro is that in the context of all of this, Alzheimer's disease is one of the most common forms of dementia, and it's associated with this loss of independence of daily functioning. So from this perspective, there is this evidence that difficulty in performing everyday activities and its consequences of reduced participation might precede MCI, mild cognitive impairment, or Alzheimer's disease, AD. So we utilize these accelerometers, thinking about measuring daily activity patterns sequentially over time may hold important potential to enhance detection for MCI and AD. Okay. Objectively measured patterns of physical activity through accelerometry really allows us to measure these detailed patterns. And as I said before, our working group led by Dr. Schrack have found that some of these patterns, such as physical activity fragmentation, which represents this breaking up of patterning. How continuous is the activity being engaged in, or how broken up is the activity being engaged in? And we find that this activity fragmentation is associated with numerous health and function outcomes. So here what we present is we show that this physical activity fragmentation is associated with mortality, this higher fragmentation associated with subsequent mortality. Further, our group has shown in a cross-sectional sense that this higher activity fragmentation is associated with lower walking speed on the y-axis. And in older adult research, as I'm sure you're aware, gait speed is a proxy of function and a hallmark indicator of living independently. On the flip side, we also want to look at the patterning of time of day, right? How does physical activity throughout the day pattern, and whether deteriorations in those patterns, those diurnal patterns, are associated with health and even cognition. We published a paper looking at high fatigability. High fatigability being defined as how fast someone reaches a state of fatigue. And we found that that was associated with deteriorating patterns of physical activity. And we really found that that difference in physical activity patterning more towards the afternoon. And then we looked at higher fatigability and fragmentation and fatigability, and we found a positive association, kind of giving us this indication that this breaking up of daily activity might be a bad thing, might be an indicator of a bad thing. So we've connected all this patterning of activity with more physical function type measures, but we really haven't looked at it in a cognitive sense. So that's really what the results here that were presented today, looking at, do any of these patterns of day-to-day physical activity differ by cognitive diagnoses? Okay, so the study aims to compare objectively measured physical activity patterns between older adults with and without MCI or AD, and we use wrist-worn accelerometry that's collected over a 24-hour period. And what we hypothesize, as you can imagine, is that older adults with the MCI or AD diagnosis have lower physical activity and more frequent rests or higher activity fragmentation throughout the day compared to counterparts without or with normal cognition. We spoke a little bit about BLSA, but just to kind of talk about it, it's an NIA-run study. It's ongoing, right? It is never ending. It's been going since 1958 or something like that. The recruitment criteria and the eligibility really end up recruiting adults who are well functioning physically and mentally. So that kind of limits generalizability, but when you find something, it's a really interesting finding. For this particular analysis, is a cross-sectional, right? But we're using data from 2015 and 2019 to identify our cross-section. We focused on older adults or adults 50 years and up who had wrist accelerometry and adjudicated cognitive diagnosis data. And our final analytic sample is 585. I'm not going to go too much into the cognitive diagnosis, but it is adjudicated by a panel of 3 physicians. Here you can see they really utilized a huge battery of neurocognitive testing along with dementia severity testing to get at MCI diagnosis or AD diagnosis. I will spend a little bit of time on the accelerometer variables because I think this is where we're trying to get at that like everyday movement and patterning within the movement. So here's an example. Let's say we have someone wearing accelerometer on their non-dominant hand. And let's say they're wearing it for 9 minutes. And that individual is walking, sits down, gets up to walk again, and then sits back down. What do we get? What do we get from the accelerometer variables? Well, first we get this total activity counts. You may have heard this term activity counts, and really they're unitless quantities of movement. And you can get them at a very detailed level. Now for us, we summarize them over minute by minute. And within those minutes, we threshold them to give it a little bit of interpretability. such that we take each minute and we look at, okay, are these activity counts at or above this validated threshold for older adults to indicate that minute is either an active minute, denoted by the green, or if it's a non-active minute, denoted by the black. And in that way, we can identify how many minutes are spent in activity or not. And these two variables up here kind of summarize the amount of activity. Now, to get to the patterning or this activity fragmentation, if you see here, you have the first 3 minutes are occurring consecutively, right? That's about. But then 2 minutes later, you have another minute. So that's how we think of continuous versus breaking up. And through that, we call it the active to sedentary transition probability. This is the probability of becoming sedentary after being active. Quite simply, mathematically, it's just the reciprocal of the average bout length collected throughout the accelerometry period. Okay, so our statistical approach, because there weren't as many MCI-AD folk who had valid accelerometry, we had to collapse it for statistical power. But what we're doing is, it's still a very important group. We're comparing between those with MCI-AD versus those with normal cognition. Statistically, we use linear regression models to estimate the differences. Each model is separated for each accelerometer metric. So you can see here, you have cognitive diagnosis as our predictor. We have our related covariates, and that should give us the difference in the accelerometer metric. For the time of day analysis, to look at times of days, we divvy up the times in 6-hour intervals. So we have like time points throughout the day, and then we can plop that into a linear mixed model to look at whether how these differences occur morning versus afternoon, or whatever these the intervals are representing. Okay, this is table 1. Wow, there's a lot here. Let me walk you through it. So each column is divvied up. So the first leftmost column is all participants, so 585. You can see those with normal cognition is a large sample of this subset. It's 549 who have normal cognition, and then MCI-AD, there's 36. So for all participants, if we go down, average age is around 75, 56% are women, almost 70% are white. These are highly educated folk, around 18 years of education. And I'm guessing you can see my mouse. The BMI is around 27, so people on average living with overweight. ApoE is around 27%, and so on. And here, importantly, usual gait speed is 1.1 meters per second at usual pace, and that's really indicative of normal functioning here. If you compare the normal cognition, the middle column, to the MCI column, what we'll see the major differences are that those with MCI-AD are older by almost 10 years, 11 years or so. Also ApoE, if you can see here, those with MCI, as we expect, have higher prevalence of the E4 allele. And then gait speed is a little bit lower, even crossing the threshold down below 1.0 meters per second here. So, and then, and I think it is important to note that the history of cardiovascular disease and stroke tend to be higher in those with MCI-AD. Okay, so that's the sample. If we look at the physical activity characteristics for all the participants and by normal and AD, sorry, I'm like moving my camera around here so I can see this. If we go to the leftmost column here, total activity counts per day is about 2.1 million counts per day in what we call valid accelerometry. What does that mean? Really, if you look at activity minutes per day, that's around 418 minutes being active versus not active, and that amounts to about 7 hours per day. Activity fragmentation is 25%. So that means when active, there's a 25% chance of transitioning into a sedentary state. We're still looking into what the clinically meaningful amount of change of activity fragmentation is. We're still looking into that. But what we think is 1% is meaningful for health and well-being. And then you can see here, normal versus AD, that those with MCI-AD tend to have lower amounts of physical activity and higher fragmentation compared to those with normal cognition. Okay, so here's table 3. This is our linear regression models. We're looking at the differences here. The first column is the beta coefficient. Our reference is normal cognition. Each 3 rows represent each accelerometer metric. So if we were to look at, oh, I'm sorry, before I get to there, if we look at total activity counts and we go to the beta coefficient, MCI versus AD, and we look at both total activity counts and activity minutes, statistically, there seems to be no difference, right? But when we get to activity fragmentation, we do find a statistical significant difference where the way we would interpret it is that those with MCI-AD have a 2% higher activity fragmentation compared to those with normal cognition, but the other 2 are similar. Okay, so before I get to the time of day analysis, here's kind of the descriptive diurnal curves between the 2 groups, where the blue line is the median for the normal cognition. The x-axis is just time of day, and then the y-axis is activity counts per minute. And then the red line is the MCI-AD. And what we can see is those MCI, the 2 lines are somewhat similar in the morning. You can see there's quite a bit of difference. Those with MCI-AD just did not collect or perform as much activity as those with normal cognition. The lines converge toward the afternoon and evening, and even cross, if you can note, towards later in the evening and closer, oh, I'm sorry, closer to midnight. So I think that was kind of interesting, and that plays into what we'll see next here in table 4, where we look at these time of day intervals. Now these are 6 hour, they're kind of crude, but let's focus on the first column. So each column is each interval, right? That 6 hour interval. So it's 12 a.m. to around 6 a.m. And what we see, what we see if we go all the way down to activity fragmentation is that those with MCI-AD actually have lower activity fragmentation compared to those with normal cognition. Now that seems backwards, but what that means is when those with MCI-AD are in an active state, they have less chance of going into a non-active state, right? 10% lower chance of going into a non-active state, which may actually indicate sleep problems because of the time of day. But then when we get to 6 a.m. To noon, and then noon to 6 a.m., you start to see these differences both by amount, right? MCI-AD having lower total activity and total activity counts. And then even in the afternoon, there's higher fragmentation, right? Those, when in an active state, there's a higher chance of going into a sedentary state here towards the afternoon. So I think that was a really interesting finding there. So just to summarize, and I know we're kind of pressed for time here. So participants with an MCI-AD diagnosis appeared to have similar overall amounts of undifferentiated physical activity engagement, but accumulated their physical activity in a more fragmented manner when compared to those with normal cognition. In the time of day analysis, that's really where we see that MCI-ADs have this earlier afternoon declines in physical activity, and then higher indications of physical activity during late evenings, perhaps indicating sleep problems. And collectively, these results suggest small but potentially important differences in patterns of daily activity that may be informative when assessing progression of cognitive change. I'll quickly go over kind of the limitations. These are important to consider, but we have this limited amount of MCI-AD, but I think with the BLSA, it's actually quite a large amount of individuals who collect accelerometry in this clinical population. It's very important to know with accelerometry, we don't capture the context of movement. What exactly are they doing? Are they sitting up, standing? So we have to keep that in our minds. And this is a cross-sectional study, so there is no temporality, right? There is reverse causation possibility. And then its limited generalizability, as BLSA tend to be healthier, older adults and well-functioning. And of course, the defined intervals are what we made up. It would be better to have these individualized levels of waking, napping, and sleeping times to really clean up the signal. But the signal does remain, and we should look further into it. And that's all I have. So here are my acknowledgments, and big thanks for the BLSA and to Jen with her grants as well. Okay, thank you.
Dr. Wanigatunga, thank you so much. Laura, I see your hand up, and I'm gonna direct you to the chat because we're running a little bit behind. I want to make sure we get everybody in there. So Amal, take a look at the chats. You've got some questions in the chat there.
Okay, yeah.
I'm gonna move on to our next presenter. This is all great, by the way. Just hopefully the take-home message is that activity is a lot more than just an average. We're gonna switch over to Dr. Rabinowitz, who hopefully can share her screen.
Hold on one second. Slideshow from start. Oops. Hold on one second. Okay. Okay. Hi, everyone. Thank you so much for attending my talk. I'm thrilled to be here and share with you a little bit about my work. All right, so today I'm going to be talking about the relationship between circadian rhythmicity and cognitive performance and decline. So it is well known that normal aging is associated with cognitive decline, which can negatively impact an individual's ability to live independently as well as quality of life. However, not everyone evidences the same rate of cognitive decline. And so it's important to identify factors that are associated with greater cognitive decline progression. So one factor that has been associated with cognitive decline is circadian rhythmicity. And so circadian rhythmicity includes several physiological processes that often cycle for over a 24-hour period. Okay, and this includes like core body temperature, melatonin, and cortisol secretion. And the circadian rhythms are under the control of the suprachiasmatic nucleus. And they are often synchronized, or circadian rest activity rhythms are synchronized by the light dark cycle through environmental cues such as light and feeding. And circadian rhythms are often derived through wrist actigraphy data. Okay. So in terms of the relationship between circadian rhythm and cognitive health, there have been a number of cross-sectional studies. So for example, cross-sectional work has found that greater intradaily variability, which is essentially greater rhythm fragmentation, which I'm going to talk about a little bit later, and lower amplitude, which is weaker rest activity rhythm strength, is associated with poorer cognitive performance on memory and language tests. There has been some longitudinal work looking at circadian rhythmicity as a predictor of cognitive decline, although there's been far less work in this area. And so studies focusing on longitudinal relations have found that those with weaker rhythm strength and greater phase delay are often at risk for heightened cognitive decline on global cognition and executive functioning tasks. Okay. But there are several limitations of the existing literature, one of which is that there's a focus on often like a global measure of cognition, but we don't really know whether or not circadian rhythms are associated with decline in specific cognitive domains, right? And there's also a failure to consider individual differences in demographic variables such as age, sex, race, and ApoE or apoprotein E4 status, which may actually modify relations between circadian rhythmicity and cognitive functioning. And the idea is that women, people of color, older age groups, and ApoE carriers, they're often at risk for mild cognitive impairment and cognitive decline. So it's possible that these folks might have might be more affected by circadian rhythmicity in terms of cognitive decline. So in the current study, we investigated cross-sectional and longitudinal associations of circadian rest activity rhythms and change over time. It changed over time in cognition in a cognitively unimpaired older adult sample. This is the same sample that Amal was talking about. And we examined whether the associations between circadian rest activity rhythms and cognitive performance and decline was moderated by age, sex, race, and ApoE allele carrier status. So I'm not really going to talk much about the BLSA sample again, because Amal just did, but the sample included 422 adults that were 50 years of age or older. They completed wrist actigraphy, cognitive assessments at 1 or more study visits, and again, they were not cognitively impaired. I have dogs in the background. I apologize in advance if you hear barking. Okay, so we looked at a number of indices of rest activity rhythms or circadian rest activity rhythms, okay? So we looked at indices of rhythm strength, one of which includes relative amplitude. What it's defined as is the normalized difference between the most active 10 hours and the least active 5 hours, which I'm going to get to in a second. Interdaily stability is the consistency of rhythm across days. Measure is the mean activity level over a 24-hour period. We also considered indices of rhythm activity. For example, we considered the total activity during the least active 5 hours and the total activity during the most active 10 hours. We also considered indices of rhythm phase delay. This includes acrophase, or the time of maximum activity counts over a 24-hour period, and then the midpoint of an individual's L5 interval, or the L5 time, the midpoint of an individual's M10 interval, or the M10 time. And last, we considered an index of rhythm fragmentation, specifically intradaily variability, which is essentially fragmentation of the rhythm relative to its 24-hour amplitude. Okay, so we assessed a number of cognitive domains, including memory, attention, executive function, and visual spatial skills. Participant demographics, age, sex, race, and years of education were obtained. Participants were genotyped. That's how ApoE E4 carrier status was ascertained. Participants also reported on their sleep medication use, depressive symptoms, and their history of conditions indicative of cardiovascular disease. BMI was also obtained, and we included these variables as covariates in our analyses. On to the results. So we found that greater rhythm strength or higher interdaily stability was associated with slower memory decline. So here I'm just talking about the main effect associations. We also found that greater activity during the least active period, or L5, was associated with slower declines in visual spatial skills. Can you actually see my slides? Okay, great, okay. All right, we also found several interactions of age with rhythm strength and fragmentation. Let me just repeat that. So we found several interactions of age with rhythm strength and fragmentation in predicting language over time. So more specifically, higher measure and less intradaily variability was associated with greater language declines at age 65, but slower language declines at age 85. So essentially, these indices were protective among older age groups, but a risk factor among younger age groups. Okay, so we also observed several race interactions. So in particular, greater amplitude, measure, and activity, and lower intradaily variability predicted slower declines in visual spatial skills among Black adults, but not white adults. We also noted several interactions with ApoE carrier status, specifically greater measure and more activity during M10 was associated with greater language declines among ApoE carrier carriers, but not non-carriers. So basically ApoE carriers were at greater risk for cognitive decline in the context of greater measure and more activity during M10. I think I missed a slide actually. So there were also several interactions with race. In particular, later L5 time was associated with lower language ability among women, but not men. In addition, later acrophase was associated with slower visual spatial skill declines in women, but not men. Okay. Excuse me 1 second. Okay. So in summary, there were several subgroups that were identified to be at risk for faster cognitive decline, depending on the circadian rest activity rhythm activity. So for example, higher measure and less intradaily variability was associated with greater language decline at younger ages compared to older age groups. And so as individuals age, it's well known that there's sort of like daily life alterations, like retirement and sort of changes in daily living that are common, which may decrease exposure to circadian synchronizing agents and alter circadian rhythmicity. Overall, though, our results suggest that interventions might be more useful among younger age groups, given that, or more useful in terms of reducing risk for cognitive decline among younger age groups. We also, as I noted, observed several interactions between amplitude, measure, and indices of activity, as well as fragmentation. So specifically, we found that lower amplitude measure activity and greater interdaily variability was associated with greater cognitive declines in visual spatial skills among Black adults, but not white adults. And so there's some research that shows that Blacks tend to have weaker rhythm strength, as well as lower circadian rhythm activity relative to whites. And this could be due to various socioeconomic influences. So for example, there's, it's thought that, differences in work schedules might, that's just differences in work schedules, but there might be a number of factors that might influence why Black adults were at risk for greater cognitive decline relative to white adults in the context of various rest activity rhythm parameters. We also found several interactions with participant sex. In particular, later acrophase was associated with slower visual spatial skills declines in women, but not men. And this is consistent with some work showing greater declines in visual spatial skills in men, but not women. The reality is there's really not much work that has investigated why this might be the case. And so there's some differences in sex hormone expression and its effects on the suprachiasmatic nucleus. Recall that circadian rest activities are regulated by the suprachiasmatic nucleus. And it's possible that differences in sex hormone expression might influence the suprachiasmatic nucleus in ways that affect risk for cognitive decline in women relative to men. As I noted before, we observed several interactions between circadian rest activity rhythms and ApoE carrier status. So specifically, we found that greater measure and more activity during M10 was associated with greater language declines in ApoE carriers, but not non-carriers. And so there's a lot of research that shows that the ApoE allele is associated with increased risk for dementia and Alzheimer's disease. And one process it's been associated with is increased accumulation of tau proteins. And increased accumulation of tau proteins have actually been associated with poor memory, which has been stronger in ApoE allele characters. There's also some research in animal models that has shown that ApoE allele variation may contribute to alterations in rest activity rhythms. However, there's so much research that needs to be done in terms of why is it that these specific rest activity rhythms are conferring risk for cognitive decline in APOE carriers, but not non-carriers. So in sum, a lot of work needs to be done specifically to determine whether or not, if you modify the rest activity rhythms among specific subgroups that were shown to be at risk, namely, for example, Black adults and APOE carriers, is that going to affect then risk for cognitive decline? I think, this area of research, this area of future research would be really, really beneficial to inform future interventions. And I'd like to thank the funding agencies that have supported this work, most notably NIA, as well as my co-authors that have made this work possible. Okay, that is it.
Thank you.
Thank you so much for listening.
Yes, Dr. Rabinowitz, that was awesome. I have a lot of questions actually for you in the chat, so take a look at those. We're going to keep going because we have about 22 minutes left for our last two speakers, and I want them to have some time to share their work. So Chengjian, why don't you share your screen now? And I believe, just wanted to say a big welcome to Dr. Wu too, who joined us at 1.
Okay, I will start sharing my screen. Oh, wait a second. Okay. So, everybody see it? Okay, so.
We're seeing your presentation mode. Did you want to?
Oh. I will just go with that.
Okay.
So yeah, hi everyone, my name is Chengjian and I am a PhD candidate in Chicago School of Molecular Engineering. And my project is using the accelerometry to predict cognitive decline among older adults without dementia. So just a little bit background introduction. So the Alzheimer's disease and related major neurocognitive disorder affect over 50 million people worldwide, and there is an increase of 10 million new cases per year. So it's necessary to develop a new sensitive predictor of early decline that can help the screening process. And so there are two tools we are using nowadays. Like first is the clinical environmental factors like age, gender, education, BMI, etc. And also we have incorporated the wearable sensors on the hip, wrist, chest data. The goal of this project is to significantly advance the prior work on forecasting early cognitive decline among older adults without dementia, but by discovering prognostic free living accelerometry pattern using not only 24, using the accelerometry data. So the way we identify whether or not there is a cognitive impairment, we are using the MoCA score, see if there is a declinement up from 1 year baseline of the MoCA score. And we are extract features from those two data to build a classifier to do the prediction. There are two samples we are using. The first is HIP, the accelerometry is a HIP1, and the study population was from the Chicago neighborhood, and the sample size is 151. And second is N-shaped wrist-worn accelerometry population that we have the sample size 793. The first set is the clinical and environmental factors we are using. As you can see here, there is other age, gender, race, education. Well, due to the time limit, I won't go to the detail, but you can check that the difference between the HIP1 data sample and the wrist-worn samples. The second is doing the cohort study. We are only analyzing the clinical and the environmental factors on whether or not the people, the subject will develop cognitive dementia after 1 year MoCA baseline, and we are analyzing the p-value on their odds ratio or effect size. and see like here I highlight two, and you can see there is some statistical significance on the age and MoCA baseline as predictors for the cognitive decline. And the second data set using is the features extracted from raw accelerometry. That there are two measurements we are using. The first is a CPM count per minute. That's normally is used to analyze the strength of accelerometry signal, and second is the vector magnitude count. That is an indicator of the amplitude deviation of accelerometry signal. So we are using some feature engineering to extract all the feasible features that are used in other literature, like the distribution of those two measures or the harmonics features of those two features. Those are all the features we are using, like, for example, like skewness, curses, entropy, and basically we are analyzing the distribution features for the CPM of VMC. And that's, we extracted in total of 98 features from the low accelerometry data. The model we use, we have tried different model, but the one work best is the gradient boosting. It's a decision tree based ensemble machine learning algorithm, not like to the linear model. And the metrics we are analyzing the performance, its accuracy, and the log score. We have three models. We have developed three models. The first is only using the clinical demographic features. The second, we have added active activity category features, which is we are count only the only the CPM of VMC that are on the above the 35% cutoff line to identify the activity level. And the third, we are including all the clinical demographic and the harmonic features together. That's the explanation of what's the AUC score, and that's the result. We have three models, and the model from 1 to 3 means the complexity is increasing gradually. So the left is the result from the heap accelerometry, and the right-hand side is the race accelerometry. For two samples, we have seen that by introducing more complex model and more information into it, we have seen a gradual improvement of the model's performance. That's the full model matrix for the heap study group, and the OX score is about 0.86 or 0.86. And the top 5 features, important features from the classifier is the VMC entropy, phosphorescence signal, skewness, and the age. And second, for the response samples, we can achieve like the OX score of 0.73. And those are the top 5 important features in this classifier. So the conclusion of our experiments is this predictive model and those set features we extracted can be used for preclinical cognitive decline generalized well over this similar accelerometry device, regardless of the location, where protocols and the follow-up time unique samples. Both samples are subject to unique pattern movement in space, but the model results are remarkably consistent over the two samples. But we have identified inferior performance of the race accelerometry among other factors may be related to the shorter wear protocol. A noteworthy point is that the robustness of the model across very unique samples. One sample representing predominant minority urban older adults with nearly 1 in 5 meeting poverty criteria, and one sample represents of the US community developing populations. And that's it. Thank you.
Thank you, Chengjian. I really appreciate your summary of that. Feel free to look at the chat. I don't think we have any questions for you yet, but feel free to put your questions in the chat and we can discuss a little bit more at the end. Dr. Wu, please feel free to share your screen now. We'll wrap up with your presentation.
No problem. Okay. Can everybody see my screen and hear me clear?
Yes.
Okay, so hi everyone, I'm Chao-Yi. I am a research associate with Oregon Health and Science University. Today I'm going to talk about the stability of indoor room transitions and their associations with myocognitive impairment. So everybody knows Alzheimer's disease. It's a devastating disease. It breaks the heart of the person and their care partners. So a lot of studies, like all the presenters presented, we try to find out whether we have measures that we can early detect, like for example, myocognitive impairment, and so that we can give them intervention to early delay this process. So we are wondering what kind of measurements out there that are potential features that can predict MCI. So in the US Food and Drug Administration 2018, their guidance has highlighted this sentence. They say cognition is meaningful, but when you measure it using conventional approach with sensitive tools directed at a particular domain, the meaningfulness of major change may not be apparent. So this is talking about traditionally, conventionally, we use paper-based measures. We ask participants to report how they feel about their cognition. Those are all great things. But we definitely need the new tools or more sensitive tools that can assess cognition in real time and objectively to understand how people are doing. So so when we think about cognition, it's kind of intuitive to think about meaningful daily routines, to think about how cognition related to your meaningful daily routines. And those daily routines, what you do every day, can be a meaningful indicator of your cognition, right? Like if your mom is cooking and it's creating a mess compared to before, she's doing pretty well and it's pretty clean, tidy, then you might think maybe something's wrong, right? So let's break down meaningful daily routines and then see whether we can assess something using technology. So in a previous paper we published, Dr. K and Miss Matake, they found out like older adults on average spend 4 hours out of home. So that means there are around 20 hours people spend at home. That's quite a lot, right? And then you think about how many hours of sleep people have, like 6 to 8 hours. So what people are doing the rest of the time, and we rarely understand what people are doing at home. We usually want to understand what they are doing outside home. So here comes to our today's topic about indoor mobility. So what do I mean by indoor mobility? So let me introduce this Mr. Smith. He went home and he went to the bathroom, washed his hands, and then he went to kitchen to make some food. And after he's in the kitchen, he went to the living room to watch some TV. And after 2 hours of TV, he wants to take a nap. So he took a nap for around maybe 1 hour, 1.5 hours. After a nap, he went to the bathroom again and still go to the living room. So this is what I mean by indoor mobility. And then obviously when people are spending, for example, 10 to 20 hours at home, people move in a home. So we're wondering if this indoor mobility can be indicative of cognitive decline. So before we evaluate their usefulness, first we need to develop algorithms to estimate indoor mobility. There are two features. First one is the frequency, and then the second one is stability. And I'll talk about that later. And M2 is to understand whether mobility stability can distinguish older adults with and without mild cognitive impairment. But let's start from M1. So how to measure indoor mobility? We, in our center, Oregon Health and Science University here, we use unobtrusive continuous in-home monitoring. So on the left-hand side, you can see the two devices. The two devices actually are passive infrared sensors. You can use stickers and put it on the wall and on the door so that you can sense, look at that, there is some sensing feature that it can sense people's movement, as well as you can put it on the door so that it can detect whether a door is open or not, so that you can estimate how much time people in the home and when they go out. And then we develop the algorithm and the below pseudocodes. Just want to show you how we estimate indoor mobility there. So there are two features. So when you look at this plot, you can see many red dots. So the larger the red dots means the more time the person spends in that room. And the smaller one, sure, it means like not much time, just some transitions across rooms. And then we develop two features. One is mobility frequency. It's daily number of room-to-room transitions at home. The other one is mobility stability. It's called interdaily stability of room transitions. I'll talk about how we calculate that shortly. So this is interdaily stability. So it quantifies the synchronization to the 24-hour light-dark cycle or rest activity rhythm. So we have two participants, day 1 and day 2, and the x-axis it's time of the day, and y-axis it's the number of room-to-room transitions counts. So you can see every day we have different activity patterns within each hour, and then it makes sense, like in the midnight, we have less room-to-room transition, but sometimes people wake up to go to bathroom or go to kitchen, so you see some activities. But in general, they are very different from day to day across hours. So we use this interdaily stability calculation, normalizing the 24 hours value from a chi-square parallelogram, and then just get basically its stability. It's a reverse. It's like it's just a flip side of variability, right? So now we got the real world data using two longitudinal cohort study, one at Oregon Health and Science University and one at Rush University. We got 139 homes, and those homes are single person homes. Single person homes, of course, because if you have two persons at home, it's hard to tease out each other's activities, right? And the number of weeks of monitoring, it's around 5,000, which is 30,000 days. And then this is a participant characteristics. Basically, people with IMCI seems like they have different age, gender proportion, and some like a number of rooms in the home. So we are going to control for that. And from the first week, we can see the first week indoor mobility frequency seems like people with MCI have more, like 7 more room transitions per day, but not significant. But in terms of indoor mobility stability, non-MCI participant, healthy cognitive participant have a higher stability than MCI participant, and it's statistically significant. And then when we look at the distribution of the two features we generated, you can see frequency in on average, people made 115 transitions per day. It's quite stunning because when you think about your own behavior, if you work from home, and can you estimate how many room-to-room transitions you made per day, you'll be surprised. It's a lot. It's more than 100 on average. And sometimes you go out and to buy something, grocery shopping, hang out with friends, it might be less. And indoor mobility stability, it's a standardized feature, so I mean it's 0.4. So M2, we want to use those two features to see whether they can distinguish people with without MCI. We use a GEE, Generalized Estimating Question Logic model, and it can take into account multiple weekly observations in estimating their standard errors. Dependent variables, it's our two features, frequency, stability. Independent variables, definitely our outcome of interest, it's a cognitive status, but also we want to control for covariates such as gait speed, age, gender, education, number of rooms at home, and time out of home actually is in there. I did not put in there. Weekly data, it's a pain severity, blueness, daylight hours. Daylight hours, interesting stuff, because actually people move less in the winter compared to summer. So we need to control for daylight hours. And then the result showed gait speed is associated with your mobility frequency. Gait speed associated with the number of transitions you made at home. And a higher speed, you made more transition. And also MCI is associated with indoor mobility stability. That means if you have cognitive impairment, your indoor mobility stability is less. And then just want to visualize it, like the relationship between mobility frequency and the gait speed, and then they are positively correlated. And an increase of a 5.3 room-to-room transition per day was associated with an increase of 10 centimeter per second gait speed. And also you can compare MCI and non-MCI in terms of their mobility stability, and showing that MCI have in general lower mobility stability. But keep in mind that this is a GEE model, which means it's on average, it did not consider individual differences, variability in this model. So conclusion is that indoor mobility, indoor room-to-room transition, they are meaningful indicators of cognition and associated with like some, it's associated with gait speed, the stability associated with cognitive status. So a pervasive sensing system deployed in homes, we think can obtrusively measure indoor activities to inform cognitive status in the older population. Thank you. And then this is my funding, and then thanks participants and my colleagues.
Thank you so much, Dr. Wu. All of this is so incredibly interesting. I know we are right at time for the demography lecture slot, but if people have a moment to stay, I just would love to take an opportunity to hear Jen's thoughts about just where do we go from here? This is an important NIA topic. We have really interesting data sets. We have really interesting measures. How do we put these together? How do we move forward into this space?
Well, that's a huge question.
Answer that in about 20 seconds.
I mean, all of this work is so amazing and important. I think we need to understand the importance of longitudinal data. Yeah. As hard as that can be to get in older adults, we need to get it. I think it also underlines the importance of remote monitoring in home and attached to the person and what we can learn. We get a lot of great data in the clinic, but we can also learn a lot of different and novel things outside of the clinic in their natural environment, moving the way they normally move. We can learn about their physical health and hopefully also their cognitive health. Mm-hmm. That's my 2 cents.
I feel like maybe if I'll add just my 1 cent compared to your 2 cents, just, you know, there's clinical applications and risk prediction, identifying people who are the most likely to decline moving forward, and they require frequent follow-up. So risk profiling. And then I think there's additional room here for potential interventions, like finding the parts of mobility that are modifiable and maybe have clinical impact or meaning. So those were just my thoughts kind of generally about some potential space. And of course, I don't know how all these work together. We have a lot of these one single metrics at a time, but there's a lot of biometrics. How do they work together? Do they measure the same things? Are they complementary? How do we pair them all together into some sort of unique bioprofile? Anyway, lots of room for growth. Does anyone else have any comments they want to share or questions?
I did wonder whether you thought about going to the participants themselves and talking to them about their lives. In other words, all of these objective measures, we measure movement from room to room and so on, you know, they look very scientific because they are very technological, but it would be really interesting to look at extreme cases and say, why do you move around all the time? Do you move around because you don't have a coffee machine in the living room? You have to go to the kitchen. Do you move around because you have your books in your bedroom? Do you move around because, you know, why do you do it? Because whenever I see these, I try to imagine myself in terms of how I would score on these things and how people I know would score. And I can see that, you know, there'd be quite different reasons for people to have these patterns. And it does seem that getting insights from the participants as to what movement means for them, they don't move because they're too tired. You know, they don't get up because if they get up, it's too much work, or they don't get up because they're well organized. I mean, these are two ways where you might not move much, and they're very different in terms of their implications. So I just wondered whether adding, in the same way that adding these technological measures to our data makes a big difference in terms of what we have. We might think again about going back and adding participant social science data to the technological data to understand what it means.
I mean, I think it's a great point. I think Amal said this maybe in his presentation. We don't know what they're doing or why they're doing. We just know that they're moving. And so understanding the context, I think, could certainly add a dimension to that we don't currently have in a lot of our work.
We're trying to go that way with this idea of ecological momentary assessment, meshing the perception with the objective measure to pull together even more precise and accurate measurements.
And I'll just point out Laura's comment, which I think is really good and kind of goes along with this, is it's not just how some individuals moving, but how is their environment supporting their movement? Like, do they have a social infrastructure that is enabling them to move more or not? Maybe it's causing harm, like, oh, I'm going to go run to the grocery store for you, Mom, so you can stay at home some more, you know, and how is that playing, you know, a role in aging and cognition? Well, it looks like we have a lot of R01s to write from this.
Really excellent.
Yeah, I really appreciate it. And I think I really also appreciate the opportunity to continue to learn all about these epidemiologic cohort studies across the US that have these complementary accelerometry data and other in-home sensor metrics that might allow us to do, you know, collaborative work. So thank you to this entire panel and Gentrack for coming back again and reliving this great experience. GSA is now calling for more symposia, so let's think about where we're going to submit this year. Thank you, everybody. Enjoy the rest of the day.